What if life in the universe didn't begin the way we've always been taught? What if the first living organisms emerged from conditions so strange, so alien to our understanding, that they defy everything we think we know about biology? Picture this: a universe where life sparks in the icy depths of interstellar clouds, or where the building blocks of existence are forged in the violent hearts of dying stars. This isn't science fiction. This is the cutting edge of astrobiology, a field that's tearing up the rulebook and rewriting the story of life itself. For centuries, we've assumed life began in warm, shallow ponds or deep sea vents on Earth. But what if our planet was just one stage in a cosmic drama that's been playing out for billions of years? The truth is, we're standing at the edge of a mystery so vast, so thrilling, it could change everything. Think about the sheer scale of the universe: trillions of galaxies, each with billions of stars, surrounded by planets we've only begun to discover. In that infinite expanse, how many times has life flickered into existence? And what did those first organisms look like? Were they fragile chains of molecules clinging to hydrothermal vents, or something far stranger, entities born from plasmas, thriving in the vacuum of space, or even riding the shockwaves of supernova? The theories we're about to explore aren't just wild guesses. They're the result of decades of research, daring experiments, and minds willing to question the impossible. From the Panspermia hypothesis to radical ideas about silicon-based life, each one offers a glimpse into a reality more extraordinary than we ever dreamed. This journey isn't just about uncovering the past, it's about redefining our future. If life can begin in ways we've never imagined, then the universe might be teeming with it, waiting for us to take the next step. Over the next 30 theories, we'll dive into the most compelling, controversial, and downright mind-bending ideas scientists have ever proposed. You'll hear about viruses older than stars, organisms that thrive in absolute zero, and even the possibility that life itself is a cosmic inevitability. So buckle up. By the time we're done, you'll never look at the night sky or the idea of life the same way again. The story of our origins is far from written. And the most astonishing chapters might still be waiting to be discovered. Let's start with one of the most captivating theories of all, Panspermia, the idea that life didn't originate on Earth at all, but instead hitched a ride here from somewhere else in the cosmos. Imagine a tiny microbe frozen in the icy depths of a comet, hurtling through space for millions of years before crashing into our planet's primordial oceans. That single organism, if the theory holds, could have been the spark that ignited all life as we know it. It sounds like something straight out of a sci-fi movie, but some of the brightest minds in science have taken this idea seriously for decades. Take Fred Hoyle and Chandra Wickramasinghe, two astronomers who boldly argued that life is far more common in the universe than we think, and that it spreads like cosmic pollen, drifting between stars on comets and asteroids. They pointed to the discovery of complex organic molecules like amino acids, floating in interstellar space and embedded in meteorites. These aren't just simple chemicals, they're the very building blocks of life, found in places where we once thought nothing could survive. And if they can form in the vacuum of space, what's stopping them from becoming something more? The evidence doesn't stop there. Some meteorites, like the famous Murchison meteorite that fell in Australia in 1969, contain molecules so intricate they mirror the ones found in living cells. Even more astonishing, certain microbes on Earth, known as extremophiles, can survive the harsh conditions of space, enduring radiation, freezing temperatures, and the vacuum of the cosmos. If these hardy organisms can cling to life in such extreme environments, why couldn't their ancestors have traveled here from another world? But Panspermia doesn't just rewrite where life began, it reshapes how we see the universe. If life can spread from one planet to another, then Earth might not be a lone oasis of biology. Instead, we could be part of an interconnected web of living worlds, all sharing the same cosmic ancestry. Maybe the same comets that delivered life to our planet also seeded Mars, Europa, or even planets in distant star systems. The implications are staggering. We might not be alone in the universe, we might be relatives. Of course, Panspermia isn't without its critics. Some argue that even if life could survive space travel, there's no definitive proof it actually happened. Others point out that the theory doesn't really answer how life began, it just moves the mystery somewhere else. But whether you believe it or not, one thing is certain: Panspermia forces us to think bigger. It challenges us to see life not as an earth-bound phenomenon, but as something that could be written into the very fabric of the cosmos. And if that's true, then the search for our origins isn't just about digging into our planet's past, it's about reaching for the stars. Now, let's plunge into the crushing depths of Earth's oceans, where sunlight never reaches and pressures could crush a submarine like a soda can. Yet here, in this alien landscape, some scientists believe life may have gotten its start. Hydrothermal vents, those towering underwater chimneys spewing superheated, mineral-rich water, are more than just geological oddities. They might be the very cradles of life. Picture a world 4 billion years ago, when Earth's oceans were a swirling chemical soup bombarded by asteroids, with no breathable oxygen in sight. In that hostile environment, these volcanic vents would have been oases of energy and stability, offering the perfect conditions for chemistry to become biology. The discovery of these vents in 1977 stunned the scientific community. Researchers aboard the submersible Alvin were exploring the Pacific Ocean floor near the Galapagos Islands when they stumbled upon something extraordinary: a thriving ecosystem completely independent of sunlight. Instead of relying on photosynthesis like nearly all surface life, these organisms thrived on chemosynthesis, drawing energy from the vents' toxic chemicals like hydrogen sulfide. Giant tubeworms, blind shrimp, and heat-loving microbes formed a food chain unlike anything seen before. But what really turned heads were the extremophile microbes living right in the scalding vent fluids, bacteria that could survive temperatures hot enough to melt lead. If life could flourish in such hellish conditions today, why couldn't it have begun there in Earth's violent infancy? The case gets even stronger when you examine the vent structure. Their porous rock formations create billions of microscopic chambers, acting like natural test tubes where organic molecules could concentrate and interact. The mix of hot and cold fluids creates natural gradients, essentially a primitive version of cellular membranes that could have helped organize early biochemistry. And the constant flow of mineral-rich water provides both raw materials and energy, solving two of the biggest puzzles about life's origins: where the building blocks came from and what powered their assembly. Some experiments even show that vent conditions can spontaneously produce key biomolecules like amino acids and lipids, lending serious credibility to this theory. But hydrothermal vents don't just offer a plausible setting for life's origins, they challenge our very definition of what life needs to exist. If organisms can thrive without sunlight or oxygen, in pressures 1,000 times greater than at sea level, then similar environments might exist across the solar system. Jupiter's moon Europa, with its suspected subsurface ocean and volcanic activity, could harbor vent ecosystems right now. Even Mars might have once had hydrothermal systems that sheltered early life. This theory doesn't just rewrite Earth's history, it opens doors to finding life in places we once thought were barren. Of course, not everyone is convinced. Critics point out that vent environments are incredibly harsh, with acidity levels that could break down complex molecules as fast as they form. And while vents explain how early life might have gotten energy, they don't fully account for how simple chemicals crossed the threshold into self-replicating systems. Still, whether or not vents were life's true birthplace, their discovery forever changed how we search for life both on our planet and beyond. They proved that biology can thrive in the most unthinkable places, turning the entire universe into a potential nursery for life. So the next time you look at the ocean, remember the greatest mysteries of our existence might not be up in the stars, but down in the crushing, fiery darkness beneath the waves. What if the secret to life's origins wasn't hidden in a primordial soup or a hydrothermal vent, but in a tiny, versatile molecule we still rely on today? Enter the RNA world hypothesis, a revolutionary idea that suggests life began with a molecule that's both a messenger and a craftsman: ribonucleic acid, or RNA. Unlike DNA, which acts like a strict librarian guarding genetic blueprints, RNA is more like a multitasking genius. It can store genetic information just like DNA, but it can also fold into intricate shapes that let it perform chemical reactions almost like a protein. This dual talent makes RNA the ultimate candidate for life's first spark, a molecule that could have copied itself without needing any other helpers. The beauty of this theory lies in RNA's simplicity. Imagine a chaotic early Earth where volcanic gases filled the atmosphere and lightning crackled across the skies. In that turbulent environment, simple molecules like cyanide and formaldehyde, chemicals we now know exist in space, could have reacted to form the building blocks of RNA. Experiments like the famous Miller-Urey study in 1953 proved these ingredients could arise spontaneously under conditions mimicking early Earth. But the real breakthrough came when scientists discovered something astonishing: RNA can indeed self-replicate. In labs, researchers have watched as strands of RNA constructed copies of themselves, no enzymes required. It's as if the molecule carries within it the seeds of its own reproduction, a whisper of the first lifelike behavior, and RNA's talents don't stop there. Some RNA molecules, called ribozymes, can chop and stitch other RNA strands, acting like molecular scissors and glue. Others can bind to small molecules, almost like a primitive form of metabolism. These aren't just chemical curiosities, they're proof that RNA could have handled the two jobs essential for life: storing information and driving the reactions needed to survive. It's like finding a Swiss Army knife in the molecular toolbox of the ancient world, capable of doing everything needed to kickstart biology. But the RNA world hypothesis isn't without its puzzles. For one, RNA is fragile. It breaks down easily, especially in the harsh conditions of early Earth. And while we've seen it self-replicate in labs, the process is messy and error-prone. Critics argue that without some kind of protective environment, like the pores of a hydrothermal vent or the surface of a clay mineral, RNA might not have stood a chance. There's also the question of where RNA's building blocks came from in the first place. Did they rain down from space or form in tidal pools under the glare of ultraviolet light? Despite these gaps, the RNA world remains one of the most compelling frameworks for understanding life's origins. It bridges the gap between chemistry and biology, showing how a molecule could cross the threshold from mere matter to something that resembles life. And the echoes of this ancient RNA world are still inside us. Every cell in your body relies on RNA to translate DNA's instructions into the proteins that build your tissues and power your thoughts. In a very real sense, we might all be descendants of a molecular pioneer that figured out how to copy itself billions of years ago. So the next time you hear about RNA in a news headline, remember, you're not just looking at a piece of biology. You're staring at the ghost of life's very first experiment. Sometimes the simplest materials hold the most extraordinary secrets. Take clay, the same humble substance we use to make pottery and bricks. What if this everyday material was the unsung architect of life itself? That's the bold claim behind the clay crystal hypothesis, a theory that suggests the first living organisms didn't emerge from a soup of chemicals alone, but were meticulously organized by the tiny, repeating structures of clay minerals. Imagine a microscopic scaffolding, invisible to the naked eye, where molecules could arrange themselves like puzzle pieces, snapping into place, guided by forces we're only beginning to understand. At the heart of this idea is the work of chemist Alexander Graham Cairns Smith, who noticed something peculiar about clay crystals. Their surfaces are riddled with intricate patterns of ridges and valleys, forming a sort of molecular blueprint. These patterns aren't random, they repeat with near-perfect precision, creating a template that could, in theory, guide the assembly of organic molecules into more complex structures. Cairns-Smith proposed that clay didn't just passively host these reactions, it actively shaped them, acting like a molecular foreman directing the construction of life's earliest building blocks. The magic lies in clay's unique properties. Many clay minerals carry slight electrical charges, allowing them to attract and hold onto molecules like amino acids or nucleotides, the very ingredients needed for life. Stacked in layers, these crystals could have concentrated these molecules in staggering quantities, creating hotspots where chemistry could take leaps forward. Even more fascinating, some clays can act as primitive catalysts, speeding up reactions that might otherwise take eons. In lab experiments, clay has been shown to help stitch together small organic molecules into longer chains, mimicking the kind of processes that could have led to proteins or genetic material. But the most compelling part of this theory isn't just clay's ability to organize molecules, it's how it might have solved one of biology's biggest chicken and egg problems. For life to begin, you need molecules that can both store information like DNA and perform work like proteins, yet each relies on the other to function. Cairns-Smith's breakthrough was realizing clay crystals could have bridged that gap. Their repeating patterns might have served as a crude form of information storage, a template that could be copied and passed down long before RNA or DNA existed. Over time, organic molecules could have learned to replicate these patterns themselves, eventually taking over the job from their mineral mentors. Critics, of course, have their doubts. While clay provides a compelling stage for life's origins, it's still unclear how the leap from mineral templates to fully organic, self-replicating systems would have happened. And not all clays are created equal. Some might hinder the very reactions they're supposed to help. But even with these unanswered questions, the clay hypothesis offers something invaluable, a plausible pathway from geology to biology. It reminds us that life might not have needed a miraculous accident, just the right set of molecular tools, patiently waiting in the dirt beneath our feet. This theory also reshapes how we search for life beyond Earth. If clay crystals could cradle life's beginnings here, why not on Mars, where vast deposits of similar minerals have been found? Or on the icy moons of the outer solar system, where clay-like materials might lurk beneath frozen oceans? The universe is full of raw materials, and if Cairns-Smith was right, some of the most ordinary ones might hold the key to the extraordinary. So the next time you see a patch of wet clay, take a closer look. You might be staring at the oldest workshop in existence, the place where chemistry first crossed into the realm of the living. Volcanic hot springs might seem like unlikely nurseries for life, steaming, acidic, and bubbling with toxic gases. Yet these turbulent landscapes could hold the key to one of biology's greatest mysteries. Picture Earth 4 billion years ago, a world of fire and chaos where volcanic activity was relentless. Amid that violence, hot springs would have been oases of stability, offering something precious, a perfect balance of energy, minerals, and protection. Unlike the deep sea vents we explored earlier, these terrestrial springs would have been exposed to the atmosphere, creating dynamic interfaces where heat, water, and gases mixed in endless combinations. It's here, in these mineral-rich pools, that the building blocks of life might have found their footing. What makes these environments so special? For starters, hot springs are natural chemistry labs. The scalding water dissolves minerals from surrounding rock, creating a broth of metals like iron, sulfur, and potassium, elements essential for life's molecular machinery. As the water cools at the edges of the spring, temperature gradients form, acting like invisible conveyor belts that shuttle molecules between hot and cold zones. This cycling could have driven reactions that wouldn't happen under steady conditions, allowing simple compounds to link into more complex chains. Some experiments even show that volcanic springs can produce key biomolecules, like the nucleotides that form RNA, when their waters mix with atmospheric gases like nitrogen and carbon dioxide. But the real magic might lie in the spring's physical structure. Many hot springs contain porous rocks. Think of them as microscopic honeycombs that could have trapped and concentrated organic molecules. These tiny chambers would have acted like primitive cells, shielding fragile reactions from the outside chaos. Meanwhile, the mineral surfaces, especially those rich in iron and sulfur, could have served as catalysts, speeding up reactions that would otherwise take millennia. It's a bit like having a built-in laboratory, complete with test tubes and reaction boosters, all provided by geology. The theory gains even more traction when you consider extremophiles, modern microbes that thrive in boiling, acidic hot springs today. Organisms like Sulfolobus, which inhabits volcanic springs at near-boiling temperatures, prove life can not only survive but flourish in these harsh conditions. If such resilient organisms exist now, why couldn't their ancient ancestors have been the first to crack the code of self-replication? Some researchers even suggest that the enzymes these microbes use to repair their DNA in extreme heat might be molecular relics from life's earliest days, a clue buried in their genes. Of course, no theory is without its challenges. Hot springs are volatile. They dry up, erupt, or shift with tectonic activity, making them unstable homes for delicate chemistry. Critics argue that without some form of protection, like fatty membranes or mineral shells, early biomolecules might have broken apart as quickly as they formed. Yet, even with these hurdles, the hot spring hypothesis offers something profound. A bridge between geology and biology, where Earth's fiery breath could have whispered life into existence. And the implications stretch far beyond our planet. If hot springs nurtured life here, similar environments on Mars, like the ancient thermal vents spotted by the Perseverance rover, might have once done the same. Even Saturn's moon Enceladus, with its geysers spewing organic, rich water into space, could be a candidate. The universe, it seems, is full of potential incubators, each waiting for the right mix of chemistry and energy to spark something extraordinary. So the next time you see steam rising from a volcanic pool, take a moment to ponder the possibility. You might be looking at the closest thing we have to a time machine, a window into life's very first moments. Cosmic radiation, the invisible, high-energy particles screaming across the universe, might seem like the last thing life would need to get started. After all, this is the same force that shreds DNA, mutates cells, and poses one of the biggest hazards for astronauts in space. But what if this seemingly destructive power was actually a creative one? Beneath the chaos of radiation lies a startling possibility. It could have been the spark that jump-started the chemistry of life. Imagine a young Earth bombarded by cosmic rays from distant supernova and our own sun, their energy transforming simple molecules into the complex building blocks of biology. It's a paradox that challenges everything we assume about life's fragility and opens up breathtaking possibilities for where else in the cosmos life might exist. The evidence for this theory comes from some of the most unexpected places. In the 1950s, a chemist named Stanley Miller, yes, the same scientist behind the famous Miller-Urey experiment, decided to test a radical idea. What if the intense radiation of Earth's early environment, far from being a barrier to life, actually helped create it? He filled a flask with simple chemicals like ammonia and methane, then blasted it with proton beams to simulate cosmic rays. The results were staggering. Not only did amino acids form, the very molecules that make up proteins, but they appeared in greater variety and quantity than in his earlier lightning-based experiments. It was as if the universe's most violent energy source had a hidden talent for molecular craftsmanship. But the surprises didn't stop there. Decades later, researchers recreated these conditions with even more precision, using particle accelerators to mimic the cosmic radiation that would have pummeled our young planet. Again and again, the same story unfolded. Radiation didn't just break molecules apart, it rearranged them into life's essential ingredients. Some experiments even produced nucleobases, the alphabet of genetic code. Suddenly, the harsh environment of early Earth looked less like a wasteland and more like a cosmic chemistry set, with radiation as its unseen alchemist. The implications ripple far beyond our own planet. If cosmic rays can forge the building blocks of life, then anywhere in the universe with similar conditions, a planet with a thin atmosphere or a moon bathed in radiation from its host gas giant could become a cradle for biology. Take Mars. Its thin atmosphere offers little protection from cosmic radiation, a fact often cited as a reason life couldn't survive there today. But what if billions of years ago that same radiation helped create life in the first place? Or Europa, where Jupiter's intense magnetic field funnels charged particles onto its icy surface. We once thought these worlds were sterile because of their radiation. Now we have to wonder if it might have been the very thing that made them fertile. Of course, skeptics raise valid concerns. Radiation is a double-edged sword. It can create biomolecules, but it can also destroy them just as quickly. Without some form of protection, like mineral shields or deep water, any delicate organic chemistry might have been snuffed out before it could take hold. And while amino acids are crucial, they're still a long way from a living cell. Still, the cosmic ray hypothesis forces us to rethink the boundaries of life's potential. It suggests that the universe doesn't just tolerate life. It might actively encourage it, using forces we once feared as tools of creation. So the next time you hear about the dangers of space radiation, remember, the same power that threatens astronauts today could have been the invisible hand that built life's first molecules. And if that's true, then the recipe for life might not be as rare as we thought, just waiting for the right burst of energy from the depths of the cosmos to set it in motion. Think of ice as nature's time capsule, not just freezing molecules in place, but actively shaping them into something greater. The cold start theory flips our assumptions about life's origins upside down, suggesting that extreme cold, far from being a barrier, might have been the perfect preservative and catalyst for life's first fragile steps. Picture Earth's early oceans dotted with icebergs or slushy patches near polar regions. At first glance, these seem like dead zones, but beneath the surface, something remarkable could have been happening. Ice's crystalline structure creates tiny channels and pockets where organic molecules would have been shielded from the harsh ultraviolet radiation bombarding the young planet, while simultaneously concentrating them like a molecular slow cooker. This isn't just speculation. Lab experiments have shown that ice can act as a surprising ally in probiotic chemistry. When water freezes, it naturally excludes impurities, forcing dissolved molecules like amino acids or nucleotides into the narrow spaces between ice crystals. There, packed tightly together, these molecules are far more likely to interact and form longer chains. Even more intriguing, the temperature fluctuations between day and night, or volcanic heat seeping through thin ice, could have created cycles of freezing and thawing. Each cycle would have acted like a reset button, allowing reactions to occur in steps too gradual for destructive forces to interfere. Scientists have replicated these conditions and watched as ice facilitated the formation of RNA-like molecules, the very stuff that might have kick-started genetics. But the real power of this theory lies in its universality. If ice provided a safe haven for life's ingredients on Earth, why not elsewhere? Look at Europa, Jupiter's enigmatic moon, where a miles-thick ice shell hides a vast subsurface ocean. Cracks in that ice, warmed by tidal forces, could be creating the same kind of protective microenvironments where molecules concentrate and react. Or consider Enceladus, where geysers blast organic, rich water into space before it falls back as icy snow. Even Mars, with its ancient glaciers and polar caps, might have once hosted similar icy nurseries. The cold start theory doesn't just expand where we look for life, it reshapes when. Icy worlds common in the outer solar system could have been incubators while Earth was still a molten hellscape. Critics, of course, point out the challenges. Ice might shield molecules from UV, but it also slows reactions to a crawl. Could life's chemistry really have advanced fast enough in what amounts to a cosmic deep freeze? And while ice preserves, it doesn't provide the energy sources that hydrothermal vents or hot springs offer. Yet even these limitations might hide unexpected advantages. Slow reactions mean fewer errors, a possible explanation for how complex molecules could have formed without falling apart. And the very scarcity of energy in icy environments might have driven early systems to become astonishingly efficient, a trait all life still shares today. What's most compelling about this idea is how it redefines habitable. We used to picture life needing warmth and liquid water. Now we see that ice itself could be the architect of biology, turning the cold void of space into a canvas for life's experiments. So the next time you see frost forming on a window or a glacier calving into the sea, remember those silent, frozen landscapes might hold the blueprint for existence itself. And if that's true, then the universe's icy frontiers could be teeming with life we've only begun to imagine. Deep beneath the ocean's surface, where sunlight never penetrates and pressure could crush steel, lies a landscape of towering chimneys spewing superheated, mineral-rich water. But these hydrothermal vents aren't just geological wonders, they might be the birthplace of life itself, thanks to a hidden partnership between chemistry and a pair of unlikely elements, iron and sulfur. German chemist Gunther Wechtersheuser saw something revolutionary in these metals. Imagine a world before proteins, before DNA, before cells, where the first sparks of metabolism flickered to life not in a watery soup, but on the jagged, reactive surfaces of iron-sulfur minerals. This is the iron-sulfur world hypothesis, a theory so bold it redefines how we picture life's very first moments. Wechtersheuser's breakthrough was realizing that pyrite fool's gold could be far more than a shiny rock. Its surface, rich in iron and sulfur, carries a slight electrical charge, making it a natural catalyst. In the vent's chaotic mix of hot water and dissolved gases, these charged surfaces could have attracted simple molecules like carbon monoxide and hydrogen sulfide, holding them in place just long enough to react. Over time, these reactions might have formed longer carbon chains, the building blocks of organic chemistry. But here's the genius part: this process wouldn't just create molecules, it would release energy. That energy, trapped and reused, could have powered even more complex reactions, creating a primitive version of metabolism long before enzymes or cells existed. It's like a molecular domino effect where each reaction sets off the next, all orchestrated by the humble chemistry of rust and sulfur. The theory gains even more traction when you look at modern biology. Deep inside your cells, in structures called mitochondria, tiny iron-sulfur clusters still play a starring role in energy production. These molecular relics are eerily similar to the mineral surfaces Wechtersheuser described, as if life never fully abandoned its ancient toolkit. Even the enzymes that drive some of our most vital metabolic reactions often have iron-sulfur cores, a fossilized clue, perhaps, to their mineral origins. It's as if biology took its first shaky steps on a scaffold of metal and stone, only later learning to build its own machinery. Lab experiments have put this idea to the test. When scientists simulate vent conditions with iron-sulfur minerals and simple gases, they've watched as these reactions produce key organic molecules, including acetic acid, a cornerstone of metabolism. Some setups even generate tiny bubbles of fatty acids, mimicking the membranes of early cells, but the most compelling evidence might come from the vents themselves. Modern hydrothermal systems host bizarre microbes that derive energy directly from iron and sulfur, living proof that this chemistry isn't just possible, it's still powering life today. Of course, no theory is perfect. Critics argue that while iron-sulfur reactions can create organic molecules, they're a long way from the self-replicating systems that define life. And the harsh conditions of deep-sea vents, scalding temperatures, crushing pressures might have shredded delicate biomolecules as fast as they formed. Yet even with these gaps, the iron-sulfur world offers something profound, a bridge between the raw geology of early Earth and the elegant complexity of biology. It suggests that life didn't need a miracle to begin, just the right combination of chemistry, energy, and time. This idea also ripples outward, reshaping how we search for life beyond Earth. If metabolism could start on mineral surfaces, then worlds like Europa or Enceladus, with their subsurface oceans and likely hydrothermal activity, might be further along the path to life than we ever imagined. Even Mars, with its ancient iron-rich rocks, could have hosted similar reactions billions of years ago. The universe, it seems, might be littered with the ingredients for life's recipe, not in some rare golden moment, but in the ordinary, grinding processes of chemistry and geology. So the next time you see a rusty nail or smell sulfur, remember, these unassuming elements might hold the secret to how existence first pulled itself from the chaos of the ancient Earth. The crackle of lightning splitting the sky isn't just a spectacle, it might have been the cosmic matchstick that lit the fuse of life. In 1953, a young chemist named Stanley Miller and his mentor Harold-Urey decided to test a radical idea. Could the violent storms of early Earth have cooked up life's ingredients? Their now iconic experiment was shockingly simple. They filled a glass flask with water and gases like methane and ammonia, mimicking Earth's primordial atmosphere, then zapped it with electrical sparks to simulate lightning. Within days, the water turned cloudy, then pink, then a deep, ominous red. When Miller analyzed the contents, he found something astonishing. Amino acids, the very molecules that build proteins, had formed spontaneously. It was as if the spark of life wasn't a metaphor, but a literal electrical jolt. But the story doesn't end there. Modern science has revived and refined this theory with twists Miller never imagined. Take volcanic lightning, a phenomenon so dramatic it sounds like something out of myth. When volcanoes erupt, the colossal friction of ash particles colliding generates violent electrical storms, far more intense than everyday lightning. Researchers now suspect these fiery displays could have been even better at forging life's building blocks than regular lightning. The reason? Volcanic plumes are packed with sulfur-rich gases, and sulfur acts like a molecular multitool, helping stitch simple molecules into complex ones. Experiments replicating these conditions have produced not just amino acids, but also nucleobases, the letters of the genetic code. Suddenly, Earth's early volcanoes weren't just destructive forces. They might have been cosmic alchemists, their eruptions weaving the first threads of biology. And the updates keep coming. Recent studies suggest the Miller-Urey experiment might have been missing a key ingredient: shockwaves. When meteorites slammed into early Earth, the colossal pressure waves ripping through the atmosphere could have supercharged chemical reactions, creating even more diverse organic molecules. Some labs have tested this by firing high-speed projectiles into gas mixtures, and the results are staggering chains of carbon molecules forming in microseconds, as if the violence of impact was the perfect catalyst. It turns out the chaos of Earth's youth wasn't an obstacle to life's origins. It might have been the essential crucible. Yet for all its elegance, the electric spark theory isn't without controversy. Critics point out that Earth's early atmosphere might not have been as rich in methane and ammonia as Miller assumed. Newer models suggest it was dominated by carbon dioxide and nitrogen, gases far less likely to produce amino acids when zapped. But here's the fascinating counterpoint. Even under these revised conditions, experiments still generate organic molecules, especially when minerals like iron or clays are present to act as helpers. And then there's the wild card: interplanetary delivery. Meteorites like Murchison, packed with amino acids, prove space is already doing this chemistry. Perhaps lightning didn't have to start from scratch, just finish the job by tweaking space-borne ingredients into something new. What's undeniable is how this idea reshapes our view of life's beginnings. If electrical energy could jumpstart biology here, why not on Venus, where thick clouds crackle with constant lightning, or on Saturn's moon Titan, where methane storms might be forging exotic versions of the same chemistry? The universe is alive with electric potential, and every bolt could be writing a recipe for life we're only beginning to decode. So the next time you watch a thunderstorm, remember, those brilliant flashes aren't just nature's fireworks, they might be echoes of the very spark that brought us into being. Imagine a droplet of oil in water, how it forms a perfect sphere, its surface a thin, flexible boundary between inside and out. Now picture that droplet not as inert chemistry, but as the very first whisper of a cell. This is the lipid bubble hypothesis, a theory that suggests life's first true breakthrough wasn't just the formation of complex molecules, but their organization into something revolutionary, tiny, self-contained worlds. Lipids, those greasy molecules that make up cell membranes today, have a remarkable property. In water, they spontaneously arrange themselves into bubbles, with their water-loving heads facing outward and their water-repelling tails tucked inside. These bubbles or vesicles aren't just passive containers. They can grow, divide, and even trap other molecules inside, creating micro-environments where chemistry could become biology. Experiments have shown that under the right conditions, like the ebb and flow of tides or the temperature gradients near hot springs, these lipid bubbles form effortlessly, as if nature itself is trying to build cells. But the real magic happens when you add RNA or other biomolecules to the mix. Some vesicles, by sheer chance, enclose strands of RNA that can self-replicate. Suddenly, you have a system where the bubble protects its fragile cargo, while the cargo, if it encodes something useful like a simple enzyme, could enhance the bubble's survival. It's a primitive version of the partnership that defines all life, a container and its instructions working in tandem. Researchers have watched this play out in labs, where lipid bubbles not only split in two but sometimes pass their molecular machinery to the next generation, like a crude form of inheritance. The implications are staggering. If lipid bubbles could form and replicate in the wild, then the leap from chemistry to biology might not have required a miracle, just the right combination of patience and physics. And the clues aren't just in test tubes. Deep in the ocean, near hydrothermal vents, scientists have found natural vesicles forming around mineral chimneys, their walls studded with the same iron-sulfur clusters that power modern metabolism. It's as if the Earth was offering up ready-made workshops for life's first experiments, yet the theory isn't without its mysteries. How did these early bubbles transition from simple dividers to sophisticated gates, selectively letting nutrients in and waste out, and what pushed them from mere survival to true reproduction, with all the complexity that entails? But every gap in our knowledge only makes the lipid bubble hypothesis more compelling. It suggests that life's origins weren't a single eureka moment, but a slow, inevitable dance of chemistry and geometry, one that might still be happening unseen on worlds far beyond our own. So the next time you see soap bubbles shimmering in the sunlight, take a closer look. You might be witnessing an echo of biology's first great invention, the art of building walls, and in doing so, creating something truly alive. The Murchison meteorite that fell to Earth in 1969 wasn't just a space rock. It was a time capsule packed with secrets. When scientists cracked it open, they found something that rewrote the rules of astrobiology: amino acids, the very building blocks of proteins embedded in its ancient grains. Not just one or two varieties, either dozens of them, some found nowhere else on Earth. This wasn't a fluke. Over the years, researchers have identified organic molecules in meteorites again and again, from sugars to nucleobases, even fragments that resemble fatty acids. These discoveries raise a staggering possibility. What if life's ingredients didn't originate on our planet at all, but rained down from the cosmos, delivered by celestial couriers billions of years ago? The chemistry inside these meteorites is astonishingly complex. Take the Murchison samples; they contain amino acids like glycine and alanine, but also exotic forms with structures never seen in terrestrial biology. Some of these molecules are even chiral, meaning they have mirror image versions, just like the molecules in our cells. The implications are profound. If space rocks can carry such sophisticated organic cargo, then the universe might be a factory for probiotic chemistry, quietly assembling life's toolkit in the void. And it's not just small molecules. In 2020, researchers found ribose, the sugar backbone of RNA in meteorites, suggesting even the scaffolding of genetics might have extraterrestrial origins. But how did these fragile molecules survive the inferno of atmospheric entry? The answer lies in the meteorite structure. Carbonaceous chondrites, the class Murchison belongs to, are like cosmic sponges, riddled with tiny pores and clay-like minerals that shield organic material from heat. Imagine a spaceship with millions of microscopic vaults, each protecting its precious cargo until it splashes down in Earth's early oceans. Some models suggest that up to half the water on our planet and countless tons of organic compounds could have arrived this way during the late heavy bombardment, a period around 4 billion years ago when asteroids pummeled the inner solar system. This theory doesn't just fill in gaps, it flips the script on life's origins. Instead of struggling to explain how simple molecules arose on a hostile early Earth, we might be looking at a universe that's already done the heavy lifting. Lab experiments show that icy grains in interstellar clouds bombarded by cosmic rays can form amino acids. Comets, those dirty snowballs wandering the solar system, are now known to harbor alcohol, formaldehyde, and other organics. Even the dust drifting between stars contains complex carbon chains. Put it all together, and you have a compelling case that Earth's first biological ingredients were imported, not homemade. Critics, of course, have their doubts. While meteorites deliver organic matter, they don't solve the mystery of how those molecules assembled into living systems. And the sheer violence of asteroid impacts enough to sterilize vast regions seems at odds with delicate chemistry. Yet new research offers a twist. Some impacts, especially those into oceans, could have created sheltered lagoons where delivered organics concentrated in warm, mineral-rich waters. It's a cosmic version of a seed landing in fertile soil, with the added kick of energy from the impact itself. The meteorite delivery theory doesn't prove Panspermia, the idea that fully formed life came from space, but it lays the groundwork. If the universe is shipping life's raw materials everywhere, then the leap from chemistry to biology might not require perfect conditions on a single planet. Instead, countless worlds could have been stocked with the same starter kit, each cooking up its own version of life. So the next time you see a shooting star, consider this. You might be watching a remnant of the same ancient delivery service that brought the ingredients for everything alive today. Along the jagged coastlines of early Earth, where crashing waves met volcanic rock, another stage for life's origins might have been quietly taking shape: tidal pools. These shallow basins, filled and drained by the relentless pull of the moon, could have been nature's perfect laboratories, harnessing the power of cycles to transform simple molecules into something extraordinary. Picture a pool at low tide, its water evaporating under the sun, leaving behind a concentrated brew of organic compounds. Then the tide rushes back in, diluting the mixture just enough to prevent stagnation, only for the cycle to repeat over and over for thousands of years. Each drying phase would have acted like a molecular pressure cooker, forcing amino acids or nucleotides to link into longer chains. Each flooding phase would have reset the system, bringing fresh ingredients and washing away waste. It's a deceptively simple process, but one that could have solved one of life's biggest puzzles. How did scattered molecules become organized, complex polymers? This idea traces its roots back to Charles Darwin himself. In a letter to a friend in 1871, he mused about life beginning in a warm little pond where heat and chemistry could work their magic. Modern science has given Darwin's poetic vision a concrete twist. Lab experiments simulating tidal pools have shown something remarkable. When researchers repeatedly wet and dry mixtures of amino acids, they form peptides, short protein-like chains. Do the same with nucleotides, and they stitch themselves into RNA-like strands. The drying phase isn't just removing water, it's bringing molecules unnervingly close, like strangers squeezed into an elevator until they have no choice but to interact. And the minerals lining these pools, like clay or volcanic rock, could have acted as catalysts, speeding up reactions that would otherwise take eons. But tidal pools offer another advantage: diversity. Unlike static environments where chemistry might hit a dead end, these dynamic basins would have created countless micro-environments. One pool might favor the formation of lipids, another might excel at knitting together nucleotides, and yet another could have been perfect for assembling metabolic cycles. Over time, waves or storms could have mixed these experiments, allowing the most successful combinations to merge and evolve. It's a bit like a vast, natural version of high-throughput screening, the same method drug companies use today to test thousands of chemical variants. Except here, the lab is the entire coastline, and the experiments run for millennia. Critics, of course, point to the challenges. Tidal pools are exposed to ultraviolet radiation, which can shred delicate organic molecules. And without some form of encapsulation, like lipid bubbles, newly formed polymers might disperse before they could act as templates for replication. But recent research offers intriguing solutions. Some minerals, like zinc sulfide common near volcanic vents, can absorb UV light, shielding molecules beneath them. And the very edges of tidal pools, where thin films of water cling to rocks, could have created semi-stable zones where early cell-like structures assembled. What's most captivating about this theory is its universality. Tidal pools aren't unique to Earth. Mars once had vast oceans, and its tidal forces, amplified by a now-lost moon, might have been even stronger. Saturn's moon Titan, with its methane lakes and shorelines, could host alien versions of the same chemistry. The universe, it seems, might be dotted with these natural laboratories, each cycling through wet and dry, concentration and dilution, chaos and order, until finally, somewhere, the right combination clicks. So the next time you see a tide pool glistening under the sun, remember, you might be looking at the closest thing we have to a cradle, not just for life on Earth, but for life anywhere. Deep beneath the ocean's surface, far from the violent chemistry of acidic hydrothermal vents, lies another kind of geological wonder, one that could rewrite the story of life's origins. Alkaline hydrothermal vents, with their towering, labyrinthine structures of porous rock, don't erupt with scalding, sulfurous plumes. Instead, they seep warm, mineral-rich fluids at a gentle pace, creating something extraordinary: natural batteries. These vents, like the Lost City field discovered in the Atlantic Ocean in 2000, are built from minerals called serpentinites, which form when seawater reacts with Earth's mantle rock. The result, a sprawling, chimney-like maze with walls riddled with microscopic pores, and inside those pores a quiet revolution might have begun. What makes alkaline vents so special is their unique energy gradient. Unlike their acidic cousins, which rely on volcanic heat, these systems harness a subtler power: chemistry. As alkaline fluids with a pH as high as 9 or 10 mix with the more acidic ocean water pH around 4, they create a natural voltage difference, almost like the positive and negative terminals of a cell. This gradient could have acted as a primitive power source, driving electrons from one compartment to another in a crude mimicry of how modern cells generate energy. Imagine these mineral walls as cosmic circuit boards, their tiny channels conducting not electricity but the very spark of life. the lost city vents discovered 20 years ago offer a breathtaking glimpse into this process. Their carbonate spires, some as tall as skyscrapers, are built by reactions between seawater and mantle rock. No magma required. The fluids seeping from them are rich in hydrogen and low in carbon dioxide, a mix that lab experiments show can spontaneously form organic molecules like formate and acetate, the building blocks of metabolism. Even more astonishing, the vent's porous structure creates billions of microscopic chambers, each a potential cradle for concentrating molecules and facilitating reactions. It's as if Earth itself engineered the perfect incubator, blending geology, chemistry, and physics into a life-generating machine. But the real magic lies in how these systems mirror modern cells. All life today uses proton gradients, differences in acidity across membranes, to power its biochemistry. Alkaline vents naturally create these gradients, with their mineral walls acting like primitive cell membranes. Some researchers argue this isn't coincidence but a fossilized clue. The first cells might not have invented this trick, they could have inherited it from the very rocks that birthed them. The vent's labyrinthine pores even resemble the folded inner membranes of mitochondria, the energy factories in our own cells. It's an eerie echo across billions of years, as if biology never fully left its mineral cradle. Critics, of course, raise questions. Alkaline vents lack the abundant sulfides and metals that drive chemistry at acidic vents, making it harder to explain how more complex molecules like nucleotides formed. And while the energy gradients are elegant, they're also delicate. Too much mixing and the system falls apart. But proponents counter that life didn't need perfection, just persistence. Over millennia, even rare successes in the vent's countless pores could have accumulated, merging into something revolutionary. What's undeniable is how this theory expands life's potential hiding places. If alkaline vents nurtured life here, similar systems might exist in the ice-covered oceans of Europa or Enceladus, where water reacts with rocky cores to produce the same chemistry. The universe, it seems, might be dotted with these silent, slow-working crucibles, each one a testament to the power of patience and the quiet genius of geology. So while acidic vents grab headlines with their infernal drama, their alkaline cousins whisper a different story. That life might not have been forged in fire, but in the gentle, relentless currents of a stone-built battery. Quantum mechanics, the bizarre rules governing the smallest particles in the universe, might seem light years away from the messy, warm world of biology. But what if life's origins didn't just depend on chemistry and energy, but on the strange, spooky behaviors of the quantum realm? Picture this: 4 billion years ago, deep in a hydrothermal vent or a tidal pool, a cluster of molecules doesn't just bump together randomly. Instead, an electron vanishes from one atom and reappears in another without crossing the space between, a trick called quantum tunneling. This isn't science fiction. Modern enzymes use this exact phenomenon to speed up reactions in your body right now. So here's the radical idea: if quantum effects power life today, could they have helped spark it into existence? The evidence starts with photosynthesis, the process plants use to turn sunlight into energy. When light hits a leaf, its energy doesn't travel in a straight line to the reaction center. It takes every possible path at once, like a ghost exploring a maze simultaneously. This quantum coherence allows near-perfect energy transfer, something classical physics can't explain. Even more astonishing, enzymes like alcohol dehydrogenase rely on quantum tunneling to shuffle hydrogen atoms around, making reactions happen millions of times faster than they otherwise would. These aren't flukes, they're fingerprints of quantum biology, proof that life harnesses the quantum world in ways we're only beginning to understand. Now rewind to life's origins. The leap from simple molecules to self-replicating systems hinges on reactions that should be too slow or improbable to matter. But quantum tunneling could change the math. Imagine a primordial RNA-like molecule trying to copy itself. A key bond needs to break, but the energy required is staggering unless the atom's nucleus tunnels through the barrier instead of climbing over it. Suddenly, an impossible reaction becomes likely. Experiments with proto-enzymes show this isn't just theoretical. Synthetic peptides in labs exhibit quantum-enhanced catalysis, suggesting early biomolecules might have exploited these effects too. Critics argue quantum states are too fragile for the noisy, wet environments where life likely began. Yet recent discoveries flip that assumption. Microscopic water droplets, like those in clouds or ocean spray, can shield quantum coherence for surprisingly long stretches. Even more compelling, certain minerals, like the iron sulfide surfaces of hydrothermal vents, can stabilize quantum effects, acting as molecular antennas that channel quantum weirdness into biological usefulness. If this theory holds, it reshapes not just life's origins but its cosmic potential. Quantum processes are universal. They don't care about temperature or pressure, only the rules of physics. A planet with the right molecular scaffolding, whether frozen or scorching, might leverage quantum tricks to kickstart biology in ways we've never imagined. So next time you see a plant glowing green in sunlight, remember, you're not just looking at chemistry, you're witnessing quantum magic, a whisper from the shadow realm that might have built the first living thing. Cosmic dust might seem like nothing more than the gritty remains of dead stars, but these tiny particles could hold the secret to life's grandest puzzle. Picture trillions of microscopic grains, each no wider than a human hair, drifting through the vast emptiness of space. On their surfaces, something extraordinary is happening. Layers of water ice, carbon monoxide, and complex organic molecules slowly accumulate, forming microscopic laboratories where chemistry experiments unfold over millions of years. These aren't just inert specks, they're cosmic workshops, and some scientists believe they might have been the staging ground for life's first act. The evidence comes from deep inside interstellar clouds, where telescopes have spotted the chemical fingerprints of life's building blocks clinging to dust grains. In the Orion Nebula, for instance, astronomers detected methanol, formaldehyde, and even ethanol molecules that, under the right conditions, can react to form sugars and amino acids. But here's the twist. In the frozen vacuum of space, these reactions should be impossibly slow. That's where the dust grains work their magic. Their surfaces provide a scaffold, holding molecules in place just long enough for cosmic rays or starlight to nudge them together. And because space is so cold, fragile compounds that would shatter on Earth can survive intact, accumulating into increasingly complex structures. It's like a slow-motion assembly line where each grain is a factory for probiotic chemistry. Lab experiments back this up. When scientists recreate space conditions in vacuum chambers, blasting icy dust analogs with ultraviolet radiation, they watch as simple molecules transform into more complex ones, even nucleobases, the letters of the genetic code. Some of these cosmic dust particles, after eons of drift, could have ended up in the molecular clouds that eventually collapsed to form solar systems. Imagine them hitching a ride on comets or asteroids, delivering their organic cargo to young planets like Earth. The Murchison meteorite, packed with extraterrestrial amino acids, might be a fossilized piece of this very process. But the story gets even stranger. Recent studies suggest these grains could do more than just carry organics. They might have actively shaped them. Dust particles in space often have a jagged, porous structure, riddled with microscopic tunnels and chambers. In simulations, these crevices act like molecular traps, concentrating ingredients and shielding reactions from destructive radiation. Some models even show that the spin of polarized light in space could have selectively favored certain molecular arrangements, potentially explaining why life on Earth prefers left-handed amino acids. It's as if the universe was subtly tilting the scales toward complexity long before planets formed. Of course, skeptics point out the gaps. While cosmic dust can create organic molecules, it's still a leap from there to self-replicating systems. And without liquid water or a stable energy source, it's unclear how these space-borne experiments could have crossed into true biology. Yet the sheer ubiquity of organic-laden dust raises a tantalizing possibility. The raw materials for life might be universal, scattered across galaxies like cosmic confetti. Whether on Earth, Mars, or a moon orbiting a distant star, the same quiet chemistry could be unfolding, one grain of dust at a time. Geothermal fields aren't just scenic landscapes of bubbling mud and steaming vents. They might be time capsules preserving the blueprint for life's earliest moments. Imagine Earth 4 billion years ago, its crust riddled with hot springs and geysers, each one a chaotic laboratory where heat, water, and minerals mixed in endless combinations. Unlike deep-sea hydrothermal vents, these terrestrial fields would have been exposed to sunlight, rain, and atmospheric gases, creating a dynamic crossroads of chemistry. Here, in the simmering broth of a geothermal pool, the ingredients for life could have found their perfect incubator, a place where energy, protection, and raw materials converged. Take Yellowstone National Park as a modern-day snapshot of this ancient world. Its hot springs teem with extremophiles microbes that thrive in near-boiling, acidic waters, their cells packed with heat-resistant proteins. These organisms, like Sulfolobus, don't just tolerate extreme conditions, they depend on them. Their enzymes, optimized for high temperatures, hint at a deep evolutionary history, one that might stretch back to a time when all life was thermophilic or heat-loving. But the real clue lies in Yellowstone's mineral deposits. The park's terraces, built layer by layer from silica and calcium carbonate, are riddled with microscopic pores. These tiny chambers could have acted as natural test tubes, concentrating organic molecules and shielding them from UV radiation while allowing just enough heat to fuel reactions. Lab experiments back up the idea. When scientists simulate geothermal conditions cycling mixtures of amino acids and nucleotides through hot and cold phases, mimicking the fluctuating temperatures of a geyser, they've watched as these molecules link into longer chains, even forming RNA-like structures. The key is the cycling itself. Heat drives reactions forward while cooling phases preserve the products, preventing them from breaking apart. Add in the mineral-rich waters, which can act as catalysts, and you've got a system where complexity emerges almost inevitably. Yet geothermal fields offer something even more tantalizing: gradients. A single hot spring might span a range of temperatures, acidity levels, and mineral concentrations across just a few feet. These gradients could have allowed different stages of life's origin to unfold side by side, simple molecules forming in the hottest zones, then drifting to cooler edges to assemble into more complex structures. It's like a factory assembly line where each station specializes in a different task, all fed by the same energy source. Critics argue that geothermal fields, like tidal pools, would have been vulnerable to UV radiation and drying. But recent discoveries in places like Chile's El Techo geyser field suggest a workaround. There, silica-rich waters form thin, translucent mineral sheets that block UV while still allowing heat and chemicals to pass through, a natural sunscreen for primordial chemistry. And in Iceland, researchers have found that geothermal steam can carry organic molecules upward, depositing them in cooler areas where they're protected yet still reactive. The implications stretch far beyond Earth. Mars's ancient volcanoes, like Olympus Mons, likely hosted vast geothermal fields. Europa's icy crust, cracked by tidal heating, might hide similar systems beneath its frozen surface. If life's blueprint was written in steaming pools and mineral scaffolds, then the universe could be dotted with copies of that same blueprint waiting for the right conditions to bring it to life. So the next time you see steam rising from a geothermal vent, think of it as more than just a geological curiosity. It might be a living relic of the moment when chemistry first crossed into the realm of biology. The young Earth was bombarded by an invisible sculptor, ultraviolet light, streaming down from the sun with an intensity we can barely imagine today. Without the protective ozone layer that now shields us, these UV rays would have been a relentless force searing the planet's surface. Yet in this apparent destruction, some scientists see a creative power, one that might have carved the first molecular steps toward life. Photochemical synthesis hinges on a radical idea that the very radiation we consider deadly could have been the chisel that shaped life's raw materials. Picture Earth's shallow ponds 4 billion years ago, their waters laced with simple molecules like hydrogen, cyanide, or formaldehyde. As UV light penetrates the surface, it doesn't just break these molecules apart, it rearranges them, injecting energy that drives new bonds to form. Experiments recreating these conditions have produced astonishing results. When scientists filled chambers with probiotic gases and exposed them to UV lamps, they watch as amino acids, sugars, and even nucleobases, the letters of genetic code, emerge spontaneously. The process mirrors what might have happened in sun-drenched tidal pools or high-altitude water droplets, where UV could act as both destroyer and architect. But the real breakthrough came when researchers realized UV light doesn't work alone. In the presence of certain minerals, its effects amplify dramatically. Zinc sulfide, a common volcanic mineral, absorbs UV and re-emits it at wavelengths that selectively favor the formation of specific biomolecules. Even more intriguing, these minerals can act as templates, aligning molecules in ways that mimic the structured complexity of life. Lab setups using these minerals have generated not just isolated compounds but entire networks of reactions, primitive metabolic cycles powered solely by light. The implications stretch beyond Earth. Mars, despite its thin atmosphere, receives intense UV radiation. If photochemistry could forge life's ingredients here, why not there? Or on exoplanets orbiting active young stars where UV fluxes dwarf our sun's output. The universe might be awash in photochemical factories, quietly assembling the pieces of life wherever starlight meets the right chemistry, yet mysteries remain. While UV can create building blocks, it also degrades them, a paradox that begs for protective mechanisms. Some researchers point to mineral shelters or lipid bubbles as potential safe havens. Others suggest the day-night cycle could have been crucial, allowing daytime synthesis and nighttime stabilization. What's undeniable is photochemistry's dual nature, a force both disruptive and creative, much like life itself. So while we slather on sunscreen to block UV's wrath, we might also owe it an unexpected debt, as the unseen hand that first whispered molecules into something greater. Black smoke vents rise from the ocean floor like industrial chimneys, spewing superheated water darkened with dissolved metals, a scorching 350 degrees Celsius, hot enough to melt lead. These towering geological formations might seem like the least hospitable places for life to begin, yet hidden in their roaring plumes of mineral-laden fluid could lie clues to biology's fiery origins. Unlike their gentler alkaline cousins, black smokers are powerhouses of raw energy, fueled by magma-heated seawater that leaches iron, sulfur, and heavy metals from Earth's crust. The result is a toxic, acidic brew that would obliterate most modern life, but for the extremophiles clinging to these vents today, it's home. Their existence hints at an astonishing possibility. Life might not have merely survived these infernal conditions. It could have been forged in them. What makes black smokers such compelling candidates? It starts with their chemistry. The plumes billowing from these vents are rich in hydrogen sulfide and dissolved metals, ingredients that, under the right conditions, can drive a primitive form of metabolism. Modern vent microbes, like those in the genus Pyrolobus, thrive here by breathing metals instead of oxygen, turning sulfides into energy in a process called chemolithotrophy. These organisms don't just tolerate extreme heat, they require it, with some species unable to reproduce below 90 degrees Celsius. Their survival strategies read like a manual for hellish environments: heat shock proteins that prevent cellular meltdown, enzymes stabilized by metal ions, and membranes built from sturdy lipids that resist disintegration. If life's earliest ancestors shared even a fraction of this resilience, black smokers would have been the ultimate proving grounds, but the vents offer more than just raw materials, they provide structure. The mineral deposits around black smokers form porous chimneys riddled with microscopic cavities. These tiny chambers act like natural reaction vessels, concentrating organic molecules while shielding them from the crushing pressures and turbulent currents outside. Inside this labyrinth, temperature gradients create natural circuits where hot fluids meet colder seawater, enabling energy harvesting reactions similar to those in modern cells. Experiments show that simulated vent conditions can drive the formation of key biomolecules, like acetic acid and pyruvate, cornerstones of metabolism. Even more compelling, iron and nickel sulfides common in vent walls can catalyze reactions that stitch carbon molecules into longer chains, mimicking the backbone of cellular biochemistry. The contrast with alkaline vents is stark, where lost city-type vents rely on gentle pH gradients and serpentinization reactions. Black smokers are brute force systems powered by volcanic heat and metal chemistry. Alkaline vents excel at producing hydrogen-rich fluids perfect for the kind of reactions that might lead to early metabolism. Black smokers, meanwhile, offer a surplus of electrons funneled through metal catalysts in ways that resemble modern respiratory chains. It's the difference between a steady drip and a lightning bolt, and it raises a tantalizing question. Did life's origins require both? Some researchers speculate that alkaline vents could have set the stage, creating stable organic building blocks, while black smokers provided the energy spikes needed to assemble them into something more complex. Yet challenges remain. The acidity of black smoker fluids can break down delicate biomolecules as fast as they form, and the vents' violent eruptions would have made them unstable homes for early life. But new discoveries keep tipping the scales. In 2021, scientists found organic coated iron particles near black smokers that mimic the structure of modern enzymes, a potential missing link between geology and biology. And on Saturn's moon Enceladus, where icy plumes hint at hydrothermal activity, similar metal-rich chemistry could be unfolding right now. So while black smokers might seem like nature's most improbable nurseries, their very extremeness could be the key. Life, it seems, doesn't just find a way, it thrives where the rules are rewritten in the crucible of chaos and heat. And if that's true, then the search for our origins and life elsewhere might need to start by looking not for comfort, but for the places where the universe burns hottest and brightest. Earth's magnetic field is more than just a navigational aid for migrating birds. It's an invisible force field that may have been life's first great guardian. Picture our planet 4 billion years ago, when the sun's ultraviolet and cosmic radiation bombarded the surface with unrelenting fury. Without protection, delicate organic molecules, the precursors to life, would have been shredded almost as quickly as they formed. But deep within Earth's molten core, swirling currents of liquid iron generated something extraordinary, a planetary-scale magnetic shield stretching thousands of miles into space. This magnetosphere didn't just deflect charged particles, it sculpted a safe zone where fragile chemistry could experiment for millennia without cosmic interference. The difference between Earth and Mars tells the story in stark relief. While both worlds once had oceans and active geology, Mars lost its magnetic field early in its history, likely as its core cooled and solidified. The result, solar winds stripped away its atmosphere over millions of years, leaving the surface exposed to radiation levels 100 times higher than Earth. Orbital images reveal the aftermath: ancient river valleys and lake beds frozen in time, their water long since boiled off into space. Any tentative steps toward complex chemistry would have been erased by the relentless particle storm. Earth's persistent magnetic field, by contrast, preserved not just our atmosphere but the very possibility of molecular evolution. This shielding did more than block radiation, it created selective pressure. Early organisms that ventured near the surface would have depended on the magnetosphere's protection, while those in deeper waters relied on physical barriers like mineral deposits or ice. Some researchers suggest this dual-layered defense allowed life to diversify, with different branches exploring varied niches without constant molecular disruption. Modern extremophiles like Deinococcus radiodurans, which can survive radiation doses lethal to humans, might be descendants of those first radiation-resistant pioneers. Their existence hints at how life could have adapted even as Earth's magnetic defenses strengthened over time, yet the magnetic field's role might go even deeper. Recent studies of deep-sea hydrothermal vents reveal an astonishing phenomenon. Some iron-sulfur minerals common in vent walls can align with Earth's magnetic field as they form, creating microscopic structures that channel electrons in organized patterns. This alignment could have provided early metabolic systems with a directional cue, like a molecular compass guiding energy flow. It's as if geology and magnetism conspired to nudge chaos toward order, offering both protection and a roadmap for life's first energy harvesting tricks. The implications for astrobiology are profound. When we scan exoplanets for habitability, the presence of a magnetic field may be just as crucial as the Goldilocks zone of liquid water. Worlds with active dynamos in their cores could be the real oases in the cosmic desert, not just briefly warm and wet, but durably shielded for the long march from chemistry to biology. As we uncover more about Earth's magnetic history, we're not just solving our own origin story. We're learning how to recognize the universe's safest nurseries, where life gets the time and shelter it needs to transform from a possibility into a certainty. Comets streak across the night sky like celestial messengers, and they might have delivered more than just fleeting beauty to Earth. Locked within their icy cores lies a treasure trove of organic compounds, from simple amino acids to complex carbon chains, all preserved in the deep freeze of space for billions of years. These cosmic snowballs, some as large as mountains, could have been the universe's original supply ships, ferrying not just the building blocks of life but also the water that filled Earth's oceans. Picture a young solar system, chaotic and violent, where comets swung wildly through the inner planets, slamming into Earth with enough force to vaporize rock, yet gentle enough to leave behind their precious cargo intact. The Rosetta mission, humanity's first spacecraft to orbit and land on a comet, gave us an unprecedented look at this process. When the probe reached comet 67P in 2014, its instruments revealed a startling truth. The comet's surface was coated with organic molecules, including glycine, the simplest amino acid. Even more compelling, the ratio of heavy water containing deuterium instead of regular hydrogen on 67P closely matched Earth's oceans, bolstering the idea that comets contributed significantly to our planet's water supply. It was a eureka, a moment for scientists, proving these icy wanderers weren't just barren chunks of ice but chemical storehouses, carrying the raw ingredients for life across the solar system. But comets offer something even more intriguing: protection. Unlike meteorites, which endure fiery plunges through the atmosphere, comets often fragment high above Earth, scattering their organic payload in a fine rain. Some models suggest this gentle delivery could have shielded fragile molecules from the destructive heat of impact, allowing them to survive intact. Experiments simulating comet impacts into early Earth's oceans show that the shock of collision might even have triggered new chemical reactions, stitching simple organics into more complex structures. Imagine a comet breaking apart midair, its icy dust settling into tidal pools or geothermal fields, where temperature cycles and mineral catalysts could further refine its cargo. The implications stretch far beyond our planet. If comets seeded Earth, why not Mars, which once had flowing rivers and lakes, or Europa, whose icy shell might hide a subsurface ocean enriched by comet strikes? The universe could be littered with similar deliveries, each one a potential spark waiting for the right conditions to ignite. And while skeptics argue that comet impacts were too violent to preserve delicate chemistry, discoveries like 67P remind us that the cosmos operates on scales we're only beginning to comprehend. Comets aren't just relics of the past, they're time capsules, offering clues to how life might arise wherever ice and organic chemistry collide. What if life didn't begin with a single molecule, but with an entire network of chemical reactions that somehow learn to sustain themselves? This is the radical idea behind autocatalytic sets, collections of molecules that don't just passively interact but actively fuel each other's creation in a closed, self-reinforcing loop. Picture a chemical version of a perpetual motion machine, where every product of one reaction becomes the starting material for another, creating a system that grows increasingly complex without any outside help. Biologist Stuart Kauffman proposed this theory in the 1970s, arguing that life's origins might not have required DNA or RNA at all. Instead, the first stirrings of biology could have been a chorus of molecules singing in perfect chemical harmony, each one keeping the others alive. The magic of autocatalytic sets lies in their simplicity. Take a hypothetical pool of early Earth molecules, amino acids, lipids, maybe a few simple sugars. Under normal circumstances, they'd float around, occasionally reacting but never amounting to much. Now imagine that by chance, one of these reactions produces a molecule that happens to speed up the creation of another molecule in the pool. That second molecule, in turn, catalyzes the formation of a third, which loops back to assist the first. Suddenly, you have a self-sustaining cycle where the whole system is greater than the sum of its parts. Kauffman showed mathematically that once a certain threshold of molecular diversity is reached, these autocatalytic sets become inevitable, not just possible but statistically unavoidable. It's like tossing a box of random gears onto a table and watching them spontaneously assemble into a working clock. Real-world experiments back up this stunning idea. In 2016, researchers created an autocatalytic set in the lab using just two simple peptides, short chains of amino acids. Astonishingly, these peptides helped synthesize each other, forming a self-replicating loop without any genetic material directing the process. Even more compelling, some modern metabolic pathways like the citric acid cycle that powers your cells still resemble autocatalytic sets, hinting at a possible evolutionary relic of this pre-RNA world. These aren't just abstract theories, they're chemical echoes of a time when life might have been defined not by information storage, but by raw, recursive chemistry. Critics, of course, raise valid questions. How did these sets transition from simple loops to the precise, information-driven systems we see in biology today? And without some form of heredity like DNA's ability to copy itself, how could they have evolved? Kauffman's answer lies in the concept of collective evolution. In a diverse enough molecular soup, small variations in reaction speeds or efficiencies would naturally lead to competition, with some sets outlasting others. Over eons, these fluctuations could drive increasing complexity, eventually stumbling upon molecules like RNA that could stabilize the system's memory. It's a bottom-up view of life's origins, where order emerges not from a single brilliant accident, but from the inevitable logic of chemistry playing out across countless interactions. This theory doesn't just rewrite life's origins, it redefines what life even is. If autocatalytic sets can arise from simple chemistry alone, then the boundary between non-living and living starts to blur. Planets with the right mix of molecules might routinely spin up these self-sustaining networks, making life less of a cosmic fluke and more of a natural phase transition. From the turbulent oceans of early Earth to the hydrocarbon lakes of Titan, anywhere chemistry has enough diversity and energy, autocatalytic whispers might be stirring. And if that's true, then the universe could be humming with proto-life, unseen and unrecognized, waiting for the right conditions to burst into full song. The same afterglow that paints our cosmic maps, the faint whisper of the Big Bang known as the cosmic microwave background, might have been more than just a relic. 14 billion years ago, this radiation wasn't the cold, dim echo we detect today. It was a searing blaze, saturating the universe with energy 1,000 times more intense than our sun. Some researchers dare to ask, could this primal light have played midwife to the first molecules of life? Picture the universe just 500,000 years after its birth, when the cosmos transitioned from a plasma soup to transparent space. The radiation bathing this newborn universe wasn't just background noise, it was a pervasive force, with wavelengths capable of breaking molecular bonds or stitching them together. At these energies, photons could have penetrated dust clouds and gas pockets, reaching the icy grains and protoplanetary surfaces where probiotic chemistry might have been stirring. Lab experiments offer tantalizing hints. When icy mixtures resembling interstellar dust are zapped with microwave radiation mimicking the early CMB, they form organic compounds like formamide, a precursor to nucleobases. It's as if the universe itself was running chemistry experiments on a cosmic scale, using light as its catalyst. Yet this theory faces a paradox. The same radiation that could drive reactions would also have been a destructive force, shredding complex molecules as quickly as they formed. The key might lie in transient safe zones, regions shielded by magnetic fields, or deep within asteroid interiors where radiation's reach was limited. Some models suggest these fluctuating environments could have created a Goldilocks scenario, just enough energy to spark reactions, but not so much as to sterilize all progress. The pulsating rhythm of early cosmic expansion might have even set the tempo, with cooling periods allowing molecules to stabilize before the next wave of energy arrived. Testing these ideas pushes the limits of imagination. Unlike hydrothermal vents or tidal pools, we can't revisit the early universe's conditions in a lab, but astrophysicists simulate them indirectly. By studying how organic molecules behave under extreme radiation in space like vacuums, or analyzing the chemistry of ancient meteorites forged in that era, we piece together a puzzle where most fragments are lost to time. The CMB's fingerprints might even linger in the molecular preferences of life itself. Some enzymes, for instance, are optimized for reactions at temperatures eerily close to those of the early universe, as if biology remembers its fiery cradle. The wildest implication: if CMB radiation truly shaped life's precursors, then the recipe isn't unique to Earth. Every corner of the cosmos was bathed in that same light, meaning the building blocks of life could have been cooking anywhere the universe provided a sheltered nook. We're left staring at the static on our cosmic TVs, wondering if it's not just noise but the faintest echo of creation's first chemistry lab. Carbon might be the superstar of life's chemistry, but it wasn't the only candidate on Earth's primordial stage. Enter silicon-carbon's understudy in the periodic table, sitting right beneath it with eerily similar properties. Both elements can form 4 bonds, allowing them to link into long chains and complex structures. So why did life bet everything on carbon and relegate silicon to rocks and computer chips? The answer lies in a cosmic tug of war between chemistry's elegance and the universe's harsh realities. Silicon's biggest strength and its fatal flaw is its love affair with oxygen. While carbon effortlessly forms and breaks bonds with hydrogen, oxygen, and itself, silicon binds so tightly to oxygen that it creates silicate minerals, locking itself away in Earth's crust. Picture a planet where silicon-based molecules try to build life, only to constantly petrify into stone. Carbon dioxide, a gas, exhales easily from living systems. Silicon dioxide, that's quartz, solid, stable, and utterly useless for dynamic biology. Yet in environments where oxygen is scarce, like the methane lakes of Titan or the high-pressure depths of gas giants, silicon might get a second chance to shine. Temperature also plays a brutal role. Carbon's bonds are strong enough to stabilize complex molecules at Earth's mild temperatures, but flexible enough to allow rapid reactions. Silicon's bonds are so heat-resistant that silicon-based reactions would crawl at a glacial pace under Earth-like conditions. But crank the heat up to volcanic extremes, and suddenly silicon springs to life. Some lab experiments show silicon chains forming spontaneously in conditions mimicking lava flows or the atmospheres of hot exoplanets. It's a reminder that if life exists in hellish environments, it might wear a very different chemical suit than we do. The most tantalizing possibility isn't on planets at all, but in space itself. In interstellar clouds where temperatures hover near absolute zero, silicon has been detected forming exotic compounds with hydrogen and nitrogen that carbon can't replicate. These molecules, fleeting on Earth, might persist for eons in the void, assembling into structures we've never imagined. Maybe life didn't just have a silicon alternative, it might have had a silicon predecessor, one that seeded carbon life before vanishing into cosmic history. For now, Earth remains a carbon-dominated world. But as we push into stranger corners of the universe, from sulfuric acid clouds to cryovolcanic moons, we can't assume biology will always follow the same rules. Silicon's story is a humbling reminder. Life as we know it might just be one variation in a symphony of possibilities, waiting to be discovered where the chemistry gets weird enough. Viruses, often dismissed as mere hijackers of life, might actually be relics of its earliest beginnings. These tiny packets of genetic material, barely more than a strand of RNA or DNA wrapped in protein, seem too simple to be ancestors. Yet their stripped-down design could be the ultimate clue to how life emerged from chemistry's chaos. Imagine a world before cells, before metabolism, where the only rule was replication. In that primordial soup, virus-like particles could have been the first molecular entities to crack the code of self-copying, a breakthrough that set the stage for everything that followed. The case for viral origins starts with their sheer minimalism. Modern viruses operate with almost nothing. Some have as few as 2 genes, compared to the thousands in even the simplest bacteria. This ruthlessly efficient design suggests an evolutionary throwback to a time when complexity wasn't just unnecessary, it was impossible. Take the hepatitis D virus, for example. It's essentially a naked loop of RNA that borrows everything else from its host, a parasitic strategy that might echo how early replicators scavenged molecules from their environment. Then there's the discovery of giant viruses like mimiviruses, which blur the line between virus and cell. Their genomes contain genes for processes like protein synthesis, traits once thought exclusive to cellular life, hinting at a lost era when viruses weren't just invaders but pioneers. But how could something that can't even reproduce without a host come first? The answer might lie in a twist. Viruses today are parasites, but their ancestors could have been self-sufficient. Some researchers propose that early virus-like replicators weren't dependent on cells because cells didn't exist yet. Instead, these molecular snippets might have replicated freely in the chemical-rich pools of early Earth, using minerals or clays as catalysts. Lab experiments support this idea. When scientists create RNA strands that can self-copy without enzymes, a feat once thought impossible, they behave strikingly like primitive viruses, mutating and competing for resources. Over time, these replicators could have evolved to specialize, with some focusing on replication while others developed protective coatings, eventually giving rise to the parasitic viruses we know today. The most tantalizing evidence might be hiding in our own DNA. Nearly 10% of the human genome consists of viral remnants, genetic hitchhikers from ancient infections. Some of these viral relics, called retrotransposons, still copy and paste themselves into our chromosomes, behaving like viruses frozen in time. Others have been co-opted for critical functions, like the syncytin proteins that help form the placenta. If viruses left such deep fingerprints on our evolution, could they have been there from the very beginning? Critics argue that viruses lack the machinery for metabolism, making them unlikely candidates for life's origin. But what if metabolism came later? In a world where raw chemical energy was abundant, replication alone might have been enough to kickstart biology. The transition from free-floating replicators to cell-based life could have been driven by competition molecules that learned to build protective bubbles. Early cells outcompeted those that didn't, eventually locking viruses into their parasitic niche. This theory doesn't just rewrite life's history, it expands where we might find it. If viruses or their precursors were the universe's first life, then places once deemed inhospitable like the acid clouds of Venus or the methane seas of Titan could harbor similar replicators, thriving without cells or even liquid water. So the next time you catch a cold, consider this: you're not just hosting a pathogen. You might be touching a living fossil, a shadow of the moment when chemistry first learned to copy itself and never stopped. At the heart of our Milky Way lies a cosmic cauldron of unimaginable energy, the galactic center, where a supermassive black hole named Sagittarius churns space-time into a frenzy. Surrounding it is a maelstrom of intense radiation, from gamma rays to X-rays, powerful enough to strip molecules apart in seconds. Yet some scientists propose this violent realm, not some quiet cosmic backwater, might have been the perfect forge for life's first sparks. The reasoning hinges on a radical twist. Chaos isn't always destructive. Sometimes it's the catalyst for breakthroughs. Picture a planet orbiting one of the ancient stars near the galactic core, bathed in relentless radiation. While this energy would have been deadly to modern life for simple molecules, it could have been a turbocharged engine for evolution. High-energy photons smashing into chemical soups might have accelerated reactions that would take millennia in calmer environments. Experiments with radiation-exposed organic compounds show something astonishing when zapped with X-rays or gamma rays. Simple molecules like formamide recombine into nucleobases, the letters of genetic code, at rates far faster than in placid conditions. It's as if the universe's most destructive force was secretly a master chemist, sculpting complexity through sheer brute force. But radiation's role goes beyond fast-tracking chemistry. Mutations, random changes in genetic material, are the raw material of evolution. Near the galactic center, radiation would have bombarded early replicators, causing errors at dizzying rates. While most mutations would have been lethal, a few might have stumbled upon innovations like better replication or energy harvesting. In this crucible, life wouldn't have had the luxury of slow refinement. It would have been forced to adapt or vanish, potentially accelerating the leap from chemistry to biology. Some extremophiles on Earth, like Deinococcus radiodurans, can survive radiation doses thousands of times stronger than what would kill a human, proving that resilience to extreme radiation isn't just possible, it's already encoded in life's playbook. The biggest obstacle isn't radiation itself, but time. The galactic center isn't just energetic, it's volatile. Supernova, gamma-ray bursts, and gravitational upheavals would have sterilized entire systems repeatedly. For life to gain a foothold, it would have needed shelters perhaps deep underground or underwater beneath miles of protective ice. On Earth, we find microbes thriving in uranium-rich mines or nuclear reactor waste, environments that echo the shielded pockets that might exist on a galactic center world. If life could anchor itself in these oases long enough to establish self-repair mechanisms, the surrounding radiation might have become less a threat and more a tool, driving diversity at a pace unimaginable in quieter neighborhoods of the galaxy. Critics, however, point to the tyranny of distance. The galactic center lies 27,000 light-years away, making it unlikely Earth's life originated there. But the theory's value isn't in explaining our own origins, it's in redefining life's potential habitats. If biology could arise in the galaxy's most hostile district, then anywhere with energy and chemistry might be a candidate. Exoplanets orbiting pulsars bathed in lethal radiation, or rogue worlds drifting through interstellar space, warmed by radioactive decay, suddenly enter the realm of possibility. The galactic center hypothesis forces us to confront a humbling truth. Life might not need a Goldilocks zone. It might only need a fighting chance, and the universe has a way of tipping the scales toward persistence. So while we may never find proof that life began near Sagittarius A, the idea reshapes our search for life elsewhere. The universe's most extreme environments, once written off as sterile, could be whispering with molecular experiments we've only begun to imagine. And if that's true, then the night sky isn't just a scattering of stars. It's a mosaic of potential biospheres, each one forged in the fires of its own unique chaos. Dark matter is the universe's most elusive architect, an invisible force shaping galaxies, yet utterly indifferent to light. We can't see it, touch it, or even detect it directly. But what if this cosmic ghost did more than sculpt the cosmos? What if it played a hand in the origins of life itself? The idea sounds like science fiction, but at the fringes of astrobiology, a daring few are asking, could dark matter's mysterious interactions have nudged chemistry toward biology in ways we're only beginning to imagine? Start with what we know, or rather, what we don't. Dark matter outweighs normal matter 5 to 1, yet it barely interacts with the stuff of stars, planets, or living things. Its particles, whatever they are, pass through Earth and Yule like shadows through a screen, but barely isn't the same as never. Some theories suggest certain dark matter particles might occasionally collide with atomic nuclei, releasing tiny bursts of energy. On early Earth, where every quantum fluctuation mattered, these rare interactions could have been wildcards, subtle jolts influencing molecular behavior. Picture a game of billiards where an invisible player occasionally nudges the balls. The game still follows known rules, but the outcome becomes unpredictable. Then there's the timing. Dark matter isn't spread evenly across the universe. Clumps of it called halos envelop galaxies, and denser strands might thread through star systems. Billions of years ago, as the solar system formed, Earth could have passed through regions where dark matter was unusually concentrated. In these pockets, those rare particle collisions would have been more frequent, perhaps enough to tip the scales for delicate probiotic reactions. Some models even propose that dark matter particles could have mediated chemical bonding, acting as temporary bridges between molecules in ways that sped up the formation of complex organics. It's a stretch, but not impossible. After all, we already know neutrinos, another elusive particle, can trigger nuclear transformations. The most radical version of this theory ventures into biology's future, not just its past. If dark matter interactions influenced life's origins, could they still be shaping evolution? Imagine a mutation caused not by radiation or copying errors, but by a dark matter particle striking DNA. The odds are vanishingly small, but over billions of years, even the improbable becomes inevitable. There's no evidence for this yet, but it's a reminder of how much we don't know about the invisible forces threading through our bodies every second. Of course, skepticism is warranted. Dark matter's properties remain hypothetical, and there's zero direct proof it affects chemistry, let alone biology. But the universe has surprised us before. Once, we thought life couldn't exist without sunlight until we found ecosystems thriving in eternal darkness. We assumed liquid water was essential until we discovered extremophiles surviving in near-pure acid. Maybe dark matter is another paradigm waiting to shift. Until we understand it fully, we can't dismiss its potential role in the grand puzzle of existence. This isn't just about rewriting life's origins, it's about redefining what's possible. If dark matter, a substance that ignores most of reality, somehow contributed to life's rise, then biology might be more deeply woven into the cosmos than we ever dreamed. The same shadowy scaffolding holding galaxies together could also, in ways we can't yet see, be holding up the story of life itself. And if that's true, then the search for our beginnings isn't just a dive into Earth's past. It's a reach into the universe's hidden dimensions. What if life on Earth didn't begin just once, but multiple times? A series of false starts and doomed experiments, with only one lineage ultimately surviving to dominate the planet. This is the spine-tingling premise of the multiple genesis hypothesis, a theory that challenges our most basic assumptions about life's origins. Instead of a single lucky accident in some ancient tidal pool or hydrothermal vent, imagine a primordial Earth where biology sparked into existence again and again, each time following a slightly different chemical blueprint. Most of these experiments would have been snuffed out by changing environments, resource competition, or sheer bad luck, leaving just one victor to inherit the world. The evidence for this idea isn't written in fossils or DNA, but in the eerie universality of life's core machinery. Every organism we've ever studied, from bacteria to blue whales, shares the same genetic code, the same basic biochemistry. This uniformity has long been taken as proof of a single origin, a last universal common ancestor, or LUCA. But what if LUCA wasn't the first life, just the last one standing? Think of it like a bustling marketplace where every stall eventually closed except one. The fact that all life today uses the same molecular toolkit doesn't mean alternatives never existed, only that they didn't survive. Some scientists point to modern extremophiles as potential echoes of these lost experiments. Deep in gold mines or acidic hot springs, researchers have found microbes with bizarre metabolic tricks so unlike anything else on Earth that they seem almost alien. Could these be the last remnants of a second genesis, clinging to existence in the margins? Even more tantalizing are synthetic biology experiments where researchers create alternative nucleic acids molecules like XNA that can store genetic information but use entirely different chemistry than DNA or RNA. If humans can engineer these in labs, why couldn't nature have stumbled upon them billions of years ago? The implications for astrobiology are profound. If life arose multiple times on Earth, a planet with a single biosphere, then the universe must be absolutely teeming with it, a cosmos where genesis is easy but survival is hard. This shifts how we search for extraterrestrial life. Instead of hunting for Earth-like biochemistries, we should be prepared to find anything from silicon-based organisms to creatures that breathe sulfuric acid. The fossil record of other planets might not show a single tree of life, but a tangled forest of false starts and dead ends. Of course, skeptics argue that without direct evidence of a second genesis, the theory remains speculative. But absence of evidence isn't evidence of absence, especially when we're searching for ghosts in 4-billion-year-old rocks. The real test may come when we explore Mars's subsurface or Europa's ocean. If we find life there with a completely different biochemical foundation, it would prove that genesis isn't a one-time fluke but a cosmic imperative. Until then, the multiple genesis hypothesis remains a humbling reminder. Life as we know it might not be special. It might just be the lucky one that got away. What if life on Earth wasn't a cosmic accident, but a deliberate act, a biological blueprint carefully planted by an intelligence beyond our world? This is the bold claim behind directed panspermia, a theory that swaps random chance for intentional design, suggesting our origins might trace back to an advanced civilization's interstellar experiment. Picture this: a spacecraft not of human making drifts toward a young Earth carrying microscopic cargo, freeze-dried microbes, or even synthetic genetic material designed to jumpstart biology on a promising planet. It sounds like the plot of a sci-fi novel, but this idea has found surprising supporters, including none other than Francis Crick, co-discoverer of DNA's double helix. Crick's involvement lends the theory an air of scientific credibility that's hard to ignore. In 1973, he and chemist Leslie Orgel published a paper arguing that the complexity of life's genetic code was statistically unlikely to arise spontaneously. Their solution, an alien civilization facing its own existential threats, might have engineered hardy microorganisms and launched them toward habitable worlds as a form of cosmic insurance. Crick wasn't claiming proof, he was highlighting a provocative possibility, one that sidestepped the gaps in conventional origin of life theories. After all, if DNA's intricate information storage seems almost too perfectly designed, could that be because it was designed just not by earthly processes? The logic of directed panspermia hinges on two compelling points. First, the universe is staggeringly old. The Milky Way formed nearly 13 billion years ago, meaning other civilizations could have had a multi-billion-year head start on us. Given that timeline, the leap from microbial engineering to interstellar seeding isn't just plausible, it's practically inevitable for any sufficiently advanced species. Second, microbes are uniquely suited for space travel. We know extremophiles can survive vacuum, radiation, and freezing temperatures, making them ideal candidates for long-duration voyages. Modern experiments have even shown bacteria surviving years on the exterior of the International Space Station, proving life can endure the harshness of space, especially if it's engineered for the purpose. But the theory's real power lies in its ability to explain anomalies. Take the genetic codes near universality. All life on Earth shares the same basic biochemical machinery, a fact often chalked up to common ancestry. But what if that uniformity reflects not a single chance origin, but a single source? Even more puzzling are molecules like molybdenum, a metal crucial for many enzymes but exceptionally rare in Earth's crust. Could its prevalence in biology be a fingerprint, a hint that our biochemical toolkit was optimized for a different planetary environment? Critics, of course, pounce on the lack of direct evidence. Without a smoking gun, an artifact, a message in our DNA, or an alien microbe with unmistakably artificial features, directed panspermia remains firmly in the realm of speculation. There's also the risk of invoking an untestable aliens did it explanation for gaps in our knowledge, a modern version of the God of the gaps fallacy. Yet even skeptics admit the theory's value lies in its audacity. It forces us to confront uncomfortable questions. How would we recognize life that was designed rather than evolved? And if we're the products of an ancient seeding event, does that make us heirs to a legacy we're only now becoming wise enough to uncover? Whether you find the idea thrilling or far-fetched, directed panspermia reshapes how we see our place in the cosmos. It turns Earth from an isolated cradle into a potential garden, one planted by hands we may never identify, but whose influence could be written into every cell of our being. And if that's true, then the search for extraterrestrial life isn't just about finding neighbors. It might be about finding our creators. Plasma, often called the fourth state of matter, is a seething, electrically charged soup of ions and electrons, found everywhere from lightning bolts to the hearts of stars. But could this chaotic medium, so alien to our watery, carbon-based existence, cradle something we might recognize as life? The idea of plasma-based organisms sounds like pure science fiction, yet some researchers argue the universe's most abundant form of matter might be hiding biology's most radical secrets. Picture a nebula light years across, its swirling tendrils not just gas and dust, but a vast, pulsating network of self-organizing structures, entities that grow, replicate, and even respond to their environment, all without a single drop of water or strand of DNA. The case for plasma life hinges on a phenomenon observed right here on Earth: plasma crystals. In lab experiments, when dust particles are suspended in plasma under the right conditions, they spontaneously arrange into intricate, lattice-like structures. These formations aren't static, they pulse, divide, and interact in ways eerily reminiscent of cellular behavior. Russian physicist V.N. Taitovich led groundbreaking studies showing these dust plasma complexes can exhibit lifelike qualities, from self-replication to rudimentary information transfer. In the microgravity of space where plasma dominates, such structures could theoretically grow far larger and more complex, evolving into something unrecognizable yet undeniably dynamic. Compare this to Earth's biology, where liquid water is the non-negotiable solvent. Water's unique properties, its ability to dissolve nutrients, facilitate chemical reactions, and maintain stable temperatures, make it the perfect cradle for life as we know it. But plasma offers its own advantages. In the depths of a nebula, where temperatures range from near absolute zero to millions of degrees, plasma's conductive nature could allow energy and information to flow at lightning speed, enabling processes far quicker than sluggish aqueous chemistry. Instead of relying on molecular bonds, plasma life might harness electromagnetic fields, using them like a nervous system to coordinate metabolism across light years of diffuse cloud. Critics, of course, dismiss the idea as fantasy. Without a clear mechanism for heredity or evolution, plasma structures, no matter how complex, remain glorified physics experiments, not true biology. Yet the universe has surprised us before. The discovery of extremophiles in boiling acid or Antarctic ice proved life thrives in places once deemed impossible. If we've learned anything, it's that biology is a shapeshifter, adapting to whatever conditions it encounters. A nebula's plasma might not resemble anything in our textbooks, but that doesn't mean it's sterile. The implications ripple beyond theory. If plasma life exists, it would redefine the hunt for extraterrestrial intelligence. Instead of scanning planets for radio signals or oxygen atmospheres, we might search nebulas for anomalous energy patterns, strange, stable vortices that defy natural turbulence. These wouldn't be little green men, but vast, drifting intelligences, their thoughts crackling along magnetic field lines, their lifespans measured in cosmic epochs. Whether such beings exist remains unknown, but the possibility forces us to confront a humbling truth. Life might not just be stranger than we imagine. It might be stranger than we can imagine. So the next time you gaze at the Orion Nebula's glowing folds, remember, you might not be looking at a stellar nursery, but a living, thinking entity, its mind woven from the fabric of space itself. What if the first living organisms didn't emerge from primordial soup or hydrothermal vents, but from lines of code? This is the mind-bending premise of the simulation hypothesis applied to life's origins. The idea that our universe's creators, whether advanced civilizations or post-human intelligences, deliberately engineered biology as part of a grand digital experiment. Imagine a scenario where the molecules of early Earth weren't just random chemical actors, but meticulously programmed variables in a cosmic simulation, each interaction fine-tuned to produce life as we know it. The implications ripple far beyond biology, forcing us to confront unsettling questions. If life was designed, who or what holds the source code? The simulation argument gains traction when you examine life's most perplexing quirks. Take the genetic code with its near-universal language across all organisms. Such uniformity could suggest a standardized blueprint, much like the repeating textures in a video game hint at programmed efficiency. Even more telling are the arbitrary choices baked into biology, like the specific 20 amino acids used to build proteins. In an unguided natural origin, we'd expect far more diversity, yet life operates with a consistency that feels almost engineered. Philosopher Nick Bostrom's famous trilemma frames the possibilities starkly. Either intelligent civilizations never reach simulation-level technology, they choose not to simulate universes, or we're almost certainly living in one. Applied to abiogenesis, this raises a haunting corollary. The first cell might not have been a lucky accident, but a deliberate subroutine. But the theory isn't just about biology, it reshapes how we interpret the universe's fundamental laws. Physics' finely tuned constants, like the strength of gravity or the mass of an electron, are so precise that even slight variations would prevent stars, planets, or chemistry from existing. Simulation proponents argue these Goldilocks values look less like cosmic luck and more like optimized parameters. In this light, life's origin stories, from RNA worlds to hydrothermal vents, aren't competing theories, but different layers of the simulation's architecture, each playing its part in a nested reality. Even quantum mechanics oddities like wave function collapse upon observation take on new meaning. Could they be processing shortcuts, the universe's way of rendering only what's necessary to maintain coherence? Critics counter with Occam's razor: why invoke unknowable programmers when chemistry suffices? Yet the simulation hypothesis does something remarkable. It bridges the gap between creationism and materialism. Unlike divine intervention, a simulated origin requires no supernatural forces, just sufficiently advanced technology operating within physical laws we might someday comprehend. It also reframes the Fermi paradox. If civilizations routinely create ancestor simulations, the cosmos might be crowded with artificial realities, not biological aliens. Our search for extraterrestrial life could be looking in the wrong dimension entirely. Ultimately, whether the simulation hypothesis holds water matters less than its power to provoke. It challenges us to see life's origins not as a solved puzzle, but as a mirror reflecting our own technological trajectory. As we inch closer to creating synthetic life in labs or running universe-scale simulations ourselves, we might be rehearsing the very process that birthed us. So the next time you marvel at DNA's elegance or the uncanny fit of Earth's conditions, consider the unsettling possibility. You're not just observing nature's handiwork, but the artifacts of a reality coded by unseen architects. The truth, as they say, might be stranger and far more meta than fiction. From the depths of hydrothermal vents to the icy vastness of interstellar space, these 30 theories aren't just competing ideas. They're pieces of a cosmic jigsaw puzzle, each revealing a different facet of life's enigmatic origins. Whether it's the electric spark of lightning forging amino acids, the molecular dance of RNA in primordial tides, or the audacious possibility that life hitched a ride here on comets, every hypothesis stretches our imagination and challenges what we thought was possible. The iron-sulfur world paints life as a biochemical offshoot of geology, while quantum biology suggests the universe itself might have nudged molecules toward complexity. Some theories, like directed panspermia, flirt with the boundaries of science fiction, while others, like the lipid bubble hypothesis, find elegance in simplicity. Yet what unites them all is a single, humbling truth. Life's beginnings are far stranger, far more inventive than we ever dreamed. This isn't just a debate about the past, it's a roadmap for the future. Every unanswered question, from the role of cosmic radiation to the potential for silicon-based life, reshapes how we search for biology beyond Earth. If life can emerge in the crushing pressure of black smoke vents or the frozen voids of space, then our definition of habitable is hopelessly narrow. The universe might be teeming with life we don't yet recognize, hiding in plain sight on worlds we've dismissed as barren. And with missions probing Europa's oceans, Mars' subsurface, and the atmospheres of distant exoplanets, we're on the brink of discoveries that could rewrite the story entirely. So which theory resonates with you? Do you see life as a cosmic inevitability, sparked by the universe's relentless chemistry, or as a fluke so rare we might be alone? Maybe you're drawn to the wildcard ideas like plasma beings or simulated origins that force us to rethink reality itself. Drop your favorites in the comments below. This conversation is just getting started. And if you're hungry for more mysteries of the cosmos, from the birth of black holes to the enigmas of dark matter, hit that subscribe button and ring the bell. The universe has kept its secrets for billions of years, but together, we're just beginning to uncover